everyone. Welcome to third annual TMT Symposium, Power of Plexing. My name is Bhavin Patel. I'll be hosting the live Q&A session for this roundtable discussion. I have all our presenters today to answer any questions specific to their talks, as well as just the general questions that you may have for them. Please use the chat function to ask questions, and I'll direct them to a specific speaker that has presented the talk. With me, I have our speaker, Kelly Staker, Chris Rose, Olga Witek, and Ryan Kelly. To kick this off, I'll go around and have a few questions for our speakers. Maybe I'll start with first questions to, maybe I'll go around and ask each one of you about your thoughts that, there are a lot of benefits to sample multiplexing for relative protein quantitation, and which one has had the largest impact on your work? Maybe I'll start with you, Kelly.
Yes, thanks. For me, the most benefit has come from dealing with missing values and how multiplexing allows you to get all of the measurements when you take one measurement. And from a biology point of view, this is so important for being able to interpret what we see, the trends that we see, because without multiplexing, differentiating the difference between a missed value and a true missing value is challenging. So that's, for me, and making stories from my data, that's been the biggest benefit to multiplexing.
Thank you, Kelly. Maybe Chris, your thoughts?
Yeah, I think much in a similar fashion, a lot of the work that we do at Genentech has been supporting our collaborators, right, who are asking biological questions. And typically those come in a fashion that really fits really well within a TMT plex, like sample multiplexing. And the ability to keep all of those within a particular experiment, like Kelly was saying, and to measure all of the proteins across those conditions, I think really helps us to answer, meaningful mechanistic biological questions. And to date, TMT has been really great in terms of the number of plexes for us, in terms of experimental size that gives us enough conditions and the different types of, spots basically to put samples to answer those questions. So that's really been the biggest thing for us. That's also the depth. We have a lot of collaborators who are interested in really low level proteins, transcription factors, other things that you can't reach until you're at like 9 or 10,000 proteins you're able to quantify. And being able to quantify at that depth across multiple samples really helps us to understand and dissect the biology better. So it's really the combination of the reduction in the missingness, the data across all the samples, but also the depth that we get from being able to fractionate beforehand and then still do the multiplex quantitation.
Thank you, Chris. Maybe Olga, any thoughts from, benefits?
Yeah, I can restate kind of the same thing, but maybe from a statistical perspective. So a lot of times in statistics, what you really care about is controlled conditions and sample size. And so multiplexing facilitates both, right? Because you measure proteins from multiple samples within the same run. So at least whatever artifacts happen in this run, they occur simultaneously in the same manner for all the samples. And also the sample size, the fact that you can now have multiple samples in one run, you can potentially have many runs, and it increases the number of biological replicas that you can have. Or alternatively, you can fractionate, and then it increases the depth of proteome coverage. So both are important, and multiplexing enables kind of working in both directions simultaneously, with the caveat, of course, that missing values are minimized within a plex, but between plexes, you have a lot of work to do to control missing values, and well, that's what we try to do in MSTANs.
Mm-hmm. Thank you, Olga. Ryan, your thoughts?
Yeah, so in the single cell proteomics space, we are, to make that a meaningful technique, we need to measure the proteomes of many cells. And I think the throughput that can be gained through multiplexing is really key for our workflow. When we look at 16, 18 plex or some of the higher order plexes that are coming out, you just can't do a label-free analysis fast enough to match the throughput that's possible through multiplexing.
Thank you, Ryan. And also, I think with the multiplexing, the precision and accuracy is also plays the big role too, right? The measurements that you're getting. And I think some of the points that Olga has also made is, you know, are you measuring what you're measuring accurately and what the precision looks like, right? With the quantitation that to achieve the higher level of quantitation, accuracy, and precision. This one question come out is, does MS stats supports hyperplexing, like TMT SILEC approach? Maybe Olga, can you answer that?
That's interesting. The answer is maybe. Conceptually, there isn't any reason why it wouldn't, or maybe with minor changes. In practice, though, we haven't seen such data. So I guess the actual answer is if there is a way for us to have example data with this type of designs, we would be happy to take a look. So maybe.
Okay. Maybe another question is, is in a past decade, there has been some major advances in LCMS instrumentation. I want to get thoughts from each one of you on any comments on how this has improved the proteomics analysis or sample multiplexing. So maybe Olga, since you're on here, I will start with you.
Okay, well, I think, again, coming from the statistics perspective, right? So better instrumentation to me, in part, means more accurate and more precise quantification, which for us translates itself into fewer interferences and therefore less artifacts of random variation. And so always when you have cleaner data, you will do better in terms of estimating fold change and sensitivity. So this is of course very important, right? And also things like dynamic range and all of that. So I think these are very positive things, but at least like for us, what it changes is really the summaries, the readouts of the statistical analysis, and then we can take those and go back and design follow-up experiments, which would use the same type of instrumentation. And we can see how experimental designs change when we move from older generation of instruments to a newer one.
Thank you, Olga. Any thoughts from Chris, your side, in terms of how the new instrumentations has improved the proteomics analysis and sample multiplexing capabilities?
Yeah, I think obviously from our talk, one of the main focuses of my own research and things that we've been implementing in the lab have been some of these intelligent data acquisition approaches. I know Kelly talked about the real-time search and the importance of that in her work. And I think for multiplexing, one of the biggest limitations for MS2-based multiplexing was interference. Obviously that was solved by MS3, but you had less signal, it took longer, there was a lot of downsides to it. But with the real-time search, I think that's made it up in our hands at least. So now we can get the same type of depth we might get with FDMS2, but we're getting more accurate quantitation with SPSMS3. And now, even with some of our internal kind of iterations of real-time search, I think we can get very specific fragment ion isolation at the SPS level, and then get even more accurate data to the point where, depending on the level of samples, like single cell, missing values start to become an issue again. But that means that the dynamic range is also actually getting larger than you might have been able to measure on previous instruments. So I think the ability to do that type of analysis, especially for multiplexing, and to kind of come up with new ways to acquire the data has been really important for us to reach out into these different types of workflows. I talked about it with the hyperplexing, coming up with new methods to kind of improve the coverage of peptides and hyperplexing. So that has been kind of the biggest thing that's impacted our research and how we run multiplex samples.
Thank you, Chris. Kelly, any thoughts from you? For the sample, how the newer instrumentations has improved the proteomics analysis and sample multiplexing. I know your workflow is combining the multiplexing with multiplexing. So, what are your thoughts are, and where do you see this is going in future?
Well, like Chris said, the smart data acquisition, I mean, to have the ability to decide what you spend your finite time on in your complex sample is really powerful. So I'm excited to see this, take off in the PTM space, because right now I think that's a struggle, and there's a huge need for that, because we battle with stoichiometry problems all the time. And if we can be smarter about what we want to pull out of our data, that's great. And we can make a lot more commentary about what's going on in our samples. And of course, the sensitivity, because now we can, with good depth, see things, pathways activated in small sample sets, or small amounts of material. So before that wasn't possible. So I think it's opening up sensitivity, and the smart acquisition are really opening up new types of samples for us to really understand.
Thank you, Kelly. Ryan, your thoughts on
yeah, so in the last few years, the ion transmission efficiency has really improved. We're using our ions more effectively due to advances in the instrumentation. And then these new mass analyzers, such as the Astral, can acquire data so much faster with fewer ions that it's really potentially game-changing. And so, it was not too many years ago when very prominent researchers were saying, well, single cell will not happen in my lifetime, right? And now, actually, the protein content from a single cell is, in some cases, a lot. You have it to play with, and it's not, we can start to contemplate smaller cells or subproteomes from these single cells, or single microbes are kind of becoming on the table now. So all of that was just not even thinkable not so many years ago. So it's quite an exciting time, and often we're just playing with these instruments, but it's the instrument manufacturers that are making a lot of this possible. And also, things are getting so fast that it's like the LC instrumentation sort of needs to catch up, in my opinion, where we've historically had to slow down our gradients to give the mass spectrometer time to chew on these ions. Now we can, we kind of need to make our separations commonly faster, because they're just the mass spectrometers so fast now.
No, thanks, Ryan. I agree. I think the mass spectrometers are much faster, but we are still working with the very similar kind of LC separation technology. And there are some improvements, but I agree with you. I think there is definitely a gap that needs to be addressed in terms of improving the throughput, also sensitivity, coupling with the more improved nano LC systems, for sure. I do want to remind our audience that please feel free to use the Q&A box to submit the questions. As you submit the questions, I'll be directing the questions to the relevant speakers. So please, I would encourage you to utilize the Q&A box to ask the questions. One question that, maybe directing to Kelly is, it was really great to see that you are kind of, once you generate the data, you are utilizing some of the bioinformatics tool to look at the functional analysis of the proteins that are differentially expressed. Just wanted to get your thoughts in terms of is there anything that's needs to be further addressed in that area of the bioinformatics, where we have a list of differentially expressed proteins, but there are tools out there that can be used, but there are more geared towards genomics or microarray data. Are there any thoughts from your side for the proteomics data for functional analysis?
Yeah, my opinion, so I'm not a statistician, so I hope I'm not misspeaking. My opinion is that for the protein data, you can get decent utility out of the genomics tools, so the expression-based enrichment tools. They work well. But where it's falling off is with the phosphorylation, because there are, PTM GSEA tools where you can look for enrichment signatures based on the phosphorylation. But I have a harder time getting robust signatures out of that, and often I reduce it down to the protein level. I have a phosphorylated protein, but then I just have to enrich based on that protein ID, and that's oversimplifying this sample. So the biggest improvement, I would say, would be in how we analyze the PTM quantitation, because it's also more complicated, because what does up mean and what does down mean? So I think there's a lot of space there. There's, of course, tools coming on all the time, and I often compare different ones, and I would like to see them give me the same answer of enrichment, but that's not always the case. Maybe other people have experience with this.
Thanks, Kelly. Any thoughts from your side, Olga? I know that kind of, doing the MStat and getting the differentially regulated proteins, but how that will connect with the biologists, right? Like if they are utilizing these kind of tools to generate or ask their biological questions, how would they address ease of use of using the bioinformatics tools for functional analysis?
Oh boy, yeah. So we are more and more working in that direction, because that's what our collaborators want to know, indeed. And so that's really an important thing. And on one hand, I can agree that some of the genomic type of tools can work, but even there, like for example, when you do gene set enrichment analysis, like simple thing like hypergeometric test, it compares, the presence of whatever interesting proteins, let's say because they're differentially abundant, against some kind of universe. So what is the universe, right? Are these all the possible proteins? Are they all the possible proteins present in this particular biological material? Are these all the proteins detectable by mass spectrometer? So there's like and all of these definitions will have implications for statistical results, right? And so I would love for all the tools to have the same answer, but there is no such thing, precisely because they're different tools and they make different assumptions, and they kind of perceive the problem slightly differently. And I think at the very least, it's really important to understand what the tool does and what the assumptions are and how the problem is approached. But beyond that, actually, I'm the first to agree that we need to do things which are specific to proteomics experiments, because there's enough specifics there, right? When somebody tells me that there's an omics, like a tool for omics, it's very hard to believe, because it means that, they're all the same, don't you worry, we can just have one tool for everything. There's no such thing. It means that people really don't appreciate enough the differences between different types of experiments and different types of molecules. And so, for example, so the type of things that we need to work on, and again, kind of agreeing with Kelly, even like mapping identifiers, right? So we have different proteoforms or different modified versions of the protein. How can we make sure that we have the right molecules actually described in these knowledge bases or in these pathways? And it's actually non-trivial at all. And it's essentially a separate research project, just kind of figuring out how to parse the intention behind like different identifiers in different types of literature. And then, of course, after that, okay, let's say we could list the molecules correctly, but what are they doing biologically? So how can we extract this information from the literature? And then how can we make use of that? So in our lab, we kind of take the view of outcomes of perturbations, saying, okay, well, now that we can understand like how different molecules quantitatively relate to each other, so what can this tell us in terms of if we perturb something upstream, what happens somewhere downstream? But again, it's easier said than done, because first of all, this identifier problem, and second, fundamentally we measure abundances, right? So either abundances of proteins or abundances of modified peptides. But in knowledge bases, not every regulation happens through the abundance, right? It happens through all kinds of other things. And so how can you actually distinguish what's happening from what we actually measure, right? And so how can we use what's measurable to infer biology? So to me, this is a much more complicated problem than what genomics well, in genomics it's complicated enough, but in proteomics, I feel like we have an extra layer of complexity, and it's really important to recognize that. And it's a really big, I think it's a research program, but as the first step, I really would like to advocate just to understand what are the assumptions that we're making and what are the limitations of these assumptions. And they are at the identifier, like Kelly was saying, okay, let's just kind of narrow down to protein IDs and ignore PTM, and all the way down to statistical assumptions, like is the statistical method or statistical test even appropriate in this context? So a lot of work to do, but I feel like we are getting better and better quality data. So having quality data is always a good thing, right? So it will always help us in method development and kind of determining things more precisely. So there's a lot to do, but I feel like we're on the right track as far as the data goes.
Cool. Yeah, thank you so much, Olga and Kelly, both of you, for your comments. My question, maybe I'll switch gear to Chris, and one of the questions that's been keep coming up with, more intelligent data acquisition strategy, right? Do you have any comments on how the AI tools or machine learning algorithms may be implemented in the future to really even take it to the next level?
Yeah, I think Thermo already has published with Devin using the spectral libraries so you could in theory predict spectral libraries and then use those within the method editor. So that's kind of a path towards being able to use some of the outputs of the machine learning algorithms on the fly. But that's something else like us and maybe other people are kicking around, is how to also implement some of those while you're actually collecting the data. Because if you're trying to use something like retention time prediction or a spectral prediction to better score peptides after analysis, post-analysis, I think one of the things that most people find when they start doing a real-time search online is that you have to be very careful about how you match online real-time search and FDR filtering to offline real-time search and FDR filtering. And if they're not matched up well, then you're going to get discrepancies in your data. In the worst case scenario, you actually identify different peptides online versus offline. That's not great, because then you might have picked SPSINs incorrectly or something like that. The best case scenario, maybe it's the same peptide and you're just not as efficient at collecting the data or filtering the data afterwards, right? So I think as the machine learning algorithms get more advanced and become more commonplace to be used post-analysis, doing that same type of thing during the analysis, if you're using something like a real-time search, will become important. So beyond that, obviously Thermo's had some other papers and other things out there, in terms of retention time prediction, improving target analysis, how many peptides you can target in a run. Those types of things I think would also have an importance in terms of data acquisition. But our group, we have a whole subgroup under Susan Klager who's doing HLA peptidomics, and that already benefits immensely from these from the rescoring algorithms, right? So then that's like kind of the next question. Folks have tried to use, say, HLA peptides and TMT. Could you use rescoring algorithms on the fly to try to help with that too? So I think there's going to be a big place for it. It's a little bit challenging, I think, to do that in real time, but it's something that we're interested in for sure.
Yep. Thank you, Chris. Maybe I'll kind of ask general questions about, I think one more question came up. Which search engine, as an example, MaxQuant or PD, would you prefer for MSStat? Is there any difference except for what search engine that, customers prefer to use? Olga, do you have any thoughts on that?
Well, first of all, I don't have any preference. We are equal opportunity collaborators, and we have converters for both MaxQuant and PDE, and also other tools such as Skyline, FracPipe, Spectronaut. There's quite a few. And so for both label-free and TMT, so at least technically speaking, it is whatever you like. But maybe looking more in depth, well, as I was saying, right, every method makes assumptions. And data processing tools, they also make assumptions. And it may actually be good to look into, sometimes it's really hard to know exactly what's happening and these assumptions are not stated, but at least empirically understand what things look like. For example, max quant tends to be quite liberal with missing values, at least for label free. And so we noticed that max quant has missing values sometimes, even if the peak is not necessarily of small intensity. So it actually really has quite a few more missing values than some other tools. It may or may not be a bad thing. It just depends on what the intention is behind these missing values. Tools such as Skyline, for example, they would have as few missing values as possible, and they would integrate the noise in the place where they expect to see a peak if they don't have it. So I would say it depends in part on your experiment, on your, like the biology that you're trying to study, on your instrumentation and how things kind of work. And also in part on, indeed, ease of use, because all of these tools, they are fairly complicated and they have many parameters. And using a tool that you're an expert in probably is a good idea. So all of these considerations, right? But as far as MS stats, we work with all of that. You can also use multiple and compare the results. And ideally, we would have similar conclusions. Again, to Kelly's point earlier, ideally, all the tools will give us the same answer. They won't, because they make different assumptions. They were originally developed with different abstractions in mind and different biology in mind. And so maybe the last point for that, what is really important, I would say, is to actually look at your data. So regardless of which tool you use and which statistical method you use, what you don't want is just generate a list of p-values and kind of forget about everything else. So for example, if you have missing values in some proteins in some conditions, ideally you would go back and actually look at the peaks, right? Look at the chromatography, kind of single line plot. So whatever your tool allows you to do, to understand that what is a likely interpretation of that. And maybe there's some kind of really silly mistake by the tool, right? Or maybe there is some complex biology and this will make a difference for what you do. So I would say technically you can do anything. In practice, there probably always the devil is in the details. And so some care is needed regardless of which tool.
Cool. Thank you, Olga. Maybe I'll ask much more like forward-looking questions that, in your opinion, what are you most excited about for the future in regards to TMT technology or quantitative proteomics? Maybe I'll start with Ryan. Your thoughts on, what do you see you're more excited about in the future in regards to the TMT-based quantitative proteomic studies?
So I know that some higher larger plexes are in the pipeline, and I think 35 plex or something like that, and that's very exciting to us. Just we've explored, as we presented, the hyperplexing, and Chris is doing great work in that as well. But having a single plex that can get to those levels without adding multiple precursors into the MS1 spectrum are quite exciting for us. That should be potentially game-changing in terms of kind of the number of cells we're able to measure per day, or samples that we're able to measure per day. So we're pretty excited about that.
Thanks, Ryan. Kelly, your thoughts on future in regards to TMT technology?
I think everyone's excited about the higher end number, but I am looking forward to a proteoform centric protein quantitation future. And to that point, there was a talk at ASMS this year from Carol Robinson's lab. They were using TMT for native mass spec to look at different membrane proteins. So I think the idea of taking multiplexing to intact protein analysis is very cool.
Cool. Thank you. Chris, any thoughts from you?
Yeah, obviously, like others, the higher plexing I think would be important. For single cell, I think it would be obviously even more important because the throughput there is a challenge, but also, in theory, the more ions that you can get into a plex, the less, say, carrier proteome, if you're using carrier proteome, you should have to use, because you don't need as much signal to get it to be detectable and selectable and to identify things. So I think that is also a good advantage there. The one thing for us that's been a big help, and I think I would like to see keep being developed, was the automation around TMT. So our group has an acceleromes that's been very useful for us in terms of preparing global proteome samples, which we do quite a bit for collaborators and within the group. And having that instrumentation where it's really just kind of plug and play has helped decrease kind of the people time quite a bit. So I think, seeing that evolve with the level of plexing, I think would be very important, because right now it's kind of capped at 16. So I think that's another thing that would be really helpful for us.
Thank you, Chris. Olga, maybe I'll close with your thoughts.
Yeah, well, so reiterating, like, control conditions and sample size, of course, it's important, but maybe let me add one more thing, which is kind of on my wish list. And I think that probably these things would be happening soon. So thinking of single cell specifically, right, and with multiplexing. So the reason why everybody's so excited about single cell is that now we can distinguish different subpopulations of cells, so that we don't have to just average everything, right? And we can say, okay, this is these cell types, and this is some other cell types, etc. And so what I'm finding challenging with this type of data is to actually use the data themselves to distinguish which cells come from which types. And so typically it would be nice to do using some kind of markers, like protein markers, which we quantify as part of the experiment, which would indicate, the cell state or cell type or something that kind of allows us to partition subpopulations. And currently it is challenging in part because of the stochasticity of quantification of proteins, at least in DDA kind of acquisition. So on my list would be multiplexing and kind of more reproducible quantitation of some important proteins. So some kind of, going closer to, like, some kind of targeted, or closer to some kind of systematic quantification of some of the important markers, which would allow us to better interpret single cell experiments. And maybe Chris and Ryan, maybe you will tell me that these things exist. I haven't seen yet, so I'm very excited and would be very interested in actually seeing some of the measurements which come out as part of the experiment and not externally, which would allow us to distinguish cell subtypes as part of multiplexing, because then we have these control conditions, but we also understand better subpopulations of cells that we are working with.
Yep. Thank you. Thank you, Olga. I think we are at the time for our session. I want to thank all of our speakers for really great presentations today. I also want to thank our audience for joining the roundtable discussions, as well as the 2 days of the TMT symposium. So thank you again, all the speakers, for great talk, and thanks, audience. And I think we'll close our roundtable discussions for the day 2 and conclude our TMT symposium, Power of Plexing for 2023. Thank you, everyone.
Thanks so much.
Thank you.
Thank you.